dia a todos. Vejam que hoje a gente está com uma audiência bastante grande, né? Isso demonstra realmente o sucesso que tem sido o programa, né? E hoje um dos temas mais relevantes, né? Um tema muito importante, muito atual. Gostaria de agradecer mais uma vez a Interfarma e todos envolvidos aí na organização do evento, né? E agradecer também a todos os speakers. Hoje nós temos 7, né? Então, eu acho que vai ser uma discussão muito importante, muito produtiva, e aí, como teremos 7, então, eu acho que vamos precisar de bastante tempo, e eu, então, encerro por aqui, agradecendo a todos os participantes, a todos os inscritos, né, de todas as áreas, não só da COPEC, mas áreas de registro, de toda a GGMed. Então, muito obrigado a todos.
Obrigada, Cláudia Osvan. Eu vou passar a palavra para a Camila, que vai falar inglês, porque ela vai explicar o ECHO para os nossos speakers. Camila, agora é você que manda.
Bom dia, pessoal. Muito obrigada pela participação. So, good morning, everyone. This is the third module of the ECHO program, an initiative co-created with ANVISA and Interfarma to expand knowledge about clinical trials. In this module, the optimization of clinical development are going to be discussed. Today, the topics related to innovative and complex studies, the adaptive studies and master protocol. I would like to thank ANVISA for the opportunity of the ECHO program and the continuous collaboration even during the pandemic situation with the additional activities that the agency is involved. I also would like to recognize the speakers of these sessions for their strong collaboration, dedication, and support in making this training. Thanks a million for your efforts. We appreciate very much your teamwork and commitment with this session, especially the speakers that are working on a bank holiday. And I also would like to thank Interfarma for the partnership and collaboration in the ECHO program. Now I will make a short introduction of the speakers in Portuguese. The chat is open during the presentations and answers will be provided in the end during the Q&A session. Então eu vou apresentar os speakers da sessão de hoje. A Chrissie Fletcher, ela é vice-presidente de desenvolvimento de estatística na GlaxoSmithKline. Ela é vice-presidente do grupo de especialistas em pesquisa clínica da FDA e lidera a subequipe focada em ensaios clínicos complexos. Chrissie é a principal representante da FDA para o grupo de trabalho do ICH E9. Nick Best é vice-presidente e chefe de estatística do Centro de Inovação de Data Science da GSK. Nick possui uma distinta carreira acadêmica em Cambridge e no Imperial College London, onde foi professora de estatística e epidemiologia. É internacionalmente reconhecida por seu trabalho em estatística bayesiana aplicada nos campos da saúde e ciências sociais. Lada Leyens é diretora de regulatórios e líder de regulatórios em inovação de estudos clínicos na Roche. Lada trabalhou em autoridades sanitárias por mais de 8 anos como revisora de ensaios clínicos e inspetora de boas práticas clínicas na Swissmedic. Na EMA, esteve no Departamento de Disciplinas Especializadas, trabalhando no procedimento centralizado. Ruchi Upmanyu é cientista principal de estatística na Roche. Ruchi trabalha em diferentes áreas terapêuticas, fornecendo conhecimentos estatísticos para o desenvolvimento de medicamentos nas fases inicial e tardia. Todd Riehl é diretor do Grupo de Ciências Clínicas na Roche. Todd é líder do desenvolvimento global para tumores agnósticos e cânceres raros na Roche, e antes de ingressar na empresa, exerceu prática clínica como farmacêutico clínico especialista na divisão de hematologia do MD Anderson por mais de 6 anos. Kaspar Rufibach é cientista especialista em estatística na Roche, realiza metodologias, fornece consultoria para projetos, treinamentos em bioestatística, trabalha com estimativas em oncologia e interage com parceiros externos na indústria, agências regulatórias e comunidade acadêmica. Jean Pintoff é diretor médico principal na Roche. Jean é hematologista e oncologista médico. Ingressou na área de desenvolvimento da empresa em 2018. As perguntas, então, podem ser realizadas por chat durante todas as apresentações e, ao final, haverá a sessão de perguntas e respostas, em que nós leremos as perguntas e os speakers darão as respostas.
Só para acrescentar...
Opa, eu só queria acrescentar que as perguntas podem ser feitas em português e depois a gente traduz.
A gente só pedir para as perguntas sejam para todos os membros da equipe, ou se tiver em inglês, ou all panelists, por favor, para que a gente possa ver. Obrigada.
Okay, go ahead.
I think we can start now.
So, thank you for the opportunity to come and participate in your training session. I'm Chrissie Fletcher, and I've been leading an initiative within FPIA on complex clinical trials. And my materials today cover some of the recommendations that we've written in a couple of white papers that we've written. Next slide. So with complex clinical trials, it is important to align on definitions. I'm sure you are aware that there are different types of complex clinical trials and increased innovation in clinical trials. Some of the key highlights that we just want to align on initially are adaptive designs. So these are planned modifications that we pre-specify that happen after the trial starts. And it's really important to make sure that the trial validity and integrity is not compromised. And that enables the trials to be more flexible and more efficient. There are the enrichment designs, which again, to increase the efficiency of drug development, we look at tailoring treatments to patients who are most likely to benefit on clinical or laboratory or genomic or proteomic factors. The master protocols are an overarching protocol designed to address multiple questions, and this may include one or more new interventions in multiple disease areas or a single disease area, and may involve looking at biomarker-defined populations or disease subtypes. Umbrella trials, this is where we think of a single disease and we're looking to study multiple interventions in the same trial. And these could be either monotherapy or in combination. A basket trial looks at looking at multiple diseases for a single new agent. Again, maybe looking at disease subtypes. And a platform trial is looking at multiple therapies in a single disease in a perpetual and open-ended manner. So treatments can drop in and drop out of the trial as they become available and when the trial can accommodate them. And finally, historical controls is where we're using existing data to be compared with new data collected in a new trial. So we're essentially borrowing information from historical data. You'll see some of these trials now summarised in the case studies as part of this training today. Next slide. So there are different categories of complex clinical trials. If you look at the bottom of the pyramid, these are broadly applied and maybe have been in place for some years. So simple adaptive trials, dropping a dose, group sequential methods, they've been around for a while. Using trial simulation to help inform designs and program level decisions, and beginning to use real world evidence to inform trial design. So these are things that are broadly applicable now across industry. And as we go up the pyramid, there might be more advanced techniques. So there are more adaptive trial designs, for example, adaptive randomizations, the platform studies that we've just talked about, looking to substitute placebo or using historical control, and looking at the use of modelling and simulation, for example, in paediatric drug development. And then there's those at the top of the pyramid, which are more trailblazing, and these are more the novel statistical methods, the more complex trial designs that you'll begin to see in some of the case studies. And we're beginning to look at the placebo substitution used in our confirmatory trials. Next slide. So just some pictures. When we think of enrichment trials, there's a number of different types of trial designs. So this one is about biomarker stratified designs. So here we are stratifying by whether a biomarker is positive or negative. And with each of the biomarker statuses, we can balance randomization to treatment A and treatment B. Next slide. Or we have the enrichment designs where we assess a biomarker and we then randomize only the ones that are biomarker positive, and those that are biomarker negative are not part of the trial and the study objectives. And then the biomarker strategy design type C, where again we assess the biomarker, we randomize, and within the biomarker status, if they're positive, they may get randomized to treatment A, negative to treatment B, but we also have a concurrent control arm who are also randomized to treatment B. So that you can see there are different types of enrichment designs, all of which are trying to address slightly different questions. Next slide. And when we think of adaptive trials, again, there are very different types of parts of the trial that can be adapted. So it might be relating to the randomization. So as we continue through the trial, we might change the ratio of randomization rather than in a fixed design, which is 1-to-1. It might be that we skew the randomization to the therapies or doses that are more likely to be more successful, and that might lead to a reduced sample size required. We might look at adaptive dose finding, again, using probability of success to select doses. We have the group sequential trial designs, again, looking at planned looks for futility or stopping early for efficacy, again, with the aim to not expose patients to ineffective treatments. We've had sample size re-estimation in our trials for some time. Again, you can do that in a blinded or unblinded manner. Most often it's because we want to check our assumptions to make sure that they still hold, and the study will retain sufficient power to assess the trial objectives. The seamless phase 2, phase 3 or phase 1, phase 3 trials, where again we try to increase the time to make important decision making in our drug development programs, so we can again accelerate the promising treatments. And finally, adaptive enrichment designs. So looking at targeting patients who are most likely to benefit, again, looking at reducing the variability if we can use, for example, biomarkers to enrich. Next slide. So just pictorially, the umbrella study, we have the single disease at the top of the umbrella. We're screening for biomarkers, and again, depending on whether they are positive or negative, we assign patients and maintain balance across the different treatments that we're trying to assess in a single trial. Next slide, please. And the basket trial, so think of the basket as looking at a single therapy, and we're looking at different diseases. So we might have different disease types, looking at different organs, but it's the same treatment that's being studied in that single trial. The use of historical control, so at the top you can see here the typical randomized controlled trial We randomise to 2 treatments, and then we're looking for the treatment effect, the delta. We might have an augmented randomised trial where we do randomise to both an active and control arm, but we do it in an unbalanced way, and we add in data from historical controls. So at the end of the day, we do have a total 1-to-1 randomisation, and we can then look for treatment effects using an augmented control arm. Or we may look at a single arm from a study and use the comparator arm, which is completely using historical data. Again, looking at enabling us to look at a treatment effect in this setting. Next slide. There's been lots of opportunities in recent years to harmonise requirements for innovation in clinical trials. I'm sure you're aware that there is ongoing discussions creating ICH E20, which is going to focus on adaptive clinical trials. And that will enable, again, all regions within ICH to harmonise on the design, conduct, analysis and interpretation of adaptive trials. I think this has been delayed because of the pandemic, but they are expecting a guideline in 2023. That's the latest information. We have ICH E6 revision 3, where again, as part of the renovation of the GCP, there's recognising the increasing diversity of clinical trial designs and different data sources. And again, that was adopted just recently in April. We have the paediatric extrapolation, ICH E11A, which also talks about trial design, statistical analysis methods to look at paediatric extrapolation, again, to help accelerate drug development and to effectively use adult data and extrapolate it for children. And there's an ongoing ICH discussion group on MIDD, or model informed drug development, which is looking at maybe developing a guideline on MIDD, and that might subsequently become an ICH guidance. And this is how we do drug development with lots of model-based drug development, and how we use modelling and simulation to support the evidence generation holistically across the life cycle. Next slide. So examples of issues that there are lots of different aspects of trial design, conduct, analysis and interpretation that can evolve with innovation in trial design. And we've tended to think of these in 3 buckets. Those that related to the scientific or technical elements, those that are relating to operational elements, and those that might link more to regulatory process or ethic committees. I won't go through all of these, but you can see there are different issues that need to be carefully considered when thinking about using a complex clinical trial. So if there are adaptations, for example, how do you maintain trial integrity? If you're not using a fixed design, how do you address multiplicity and overall type 1 error? How do you make sure that if you're looking at interim analyses, that there is no data leakage? Some of these designs do take a lot of additional complexity, and a lot of expertise is required to employ new methodology. So do you have the right expertise and resources to deliver the trial designs? What happens if there are new biomarkers that come in during your trial, and how do you choose the most appropriate comparator? It's really important operationally to think of all the trial aspects and pre-plan for those, thinking of when you're doing a multi-regional clinical trial, what might be some of the regional variations, how many amendments might be needed, what oversight is going to be needed, what happens if there is a change in some of the processes and systems that might impact the trial conduct. Often these studies where you're doing a complex design is multi-sponsor or collaborative trials, and that increases the operational aspects. And it's making sure we can maintain confidentiality, which may have contractual issues. And on the regulatory and ethics side, we understand there are regulatory uncertainties. There are questions about the acceptability of data, especially in where we're looking to use historical control. How much experience and expertise have the regulators been exposed to, and how complex are protocols, and how do we explain that in a very careful and deliberate way? It might increase workload at the agency to review these studies. There's lots of concerns about transparency and making sure that we're meeting our obligations. And in the platform studies, when does a trial end is of a concern, in particular to ethics committees. Next slide. So there are numerous challenges, and we understand those. And what we're trying to think of is, how do we work together to address some of these challenges? So we are looking to understand how we bridge the understanding, the expertise, how do we collaborate in developing new guidance? If industry were to prioritise a complex trial, is that going to be accepted as evidence for both the regulators and any reimbursement agencies? How can we come together to fix some of the challenges and work on solutions together? What are emerging best practices that we are seeing, both from all the stakeholder views, including the patient's perspective? How do patients feel about some of these complexities in these new trials? And how do you also continue to accelerate and drive innovation, as well as meeting the needs? And we've learned a lot from the COVID-19 pandemic in that regard. Next slide. So we have come up with some potential solutions that we think it is really important to build technical capabilities and capacities. By collaborating between different stakeholders, it really does help to identify what the issues are and to allow the conversations to be had in a timely fashion. So we want, sponsors would like to get timely advice and opportunities to engage early in the process as much as possible, so we can understand what are the concerns by different stakeholders. But ultimately, we need global approaches, because most of the trials that we run are global in nature and international in nature. So to serve all stakeholders, we need to come together and harmonise. Next slide. So my final slide is innovation and complex trials is a change, both for patients, for sponsors, regulators and reimbursement agencies. But we're all here to try to make the best decision at the earliest time in the most efficient manner. So we've identified that we do need to prioritise education. We need as much discussion as we can have as part of the process. And we need acceptability for the data. So we need to make sure that any evidence that we generate is going to meet the needs for the regulatory decision making that is required. And hopefully we feel it's going to lead to better treatments for patients, requiring robust evidence that will be needed for the different decision makers. So I hope that was a sort of a short introduction to complex trials aligned on definitions. Now we're going to show you some case studies which we hope will illustrate some of these points.
Thank you, Chrissie. Next slide.
Sorry, Lada, just a comment, an information for the public. Só para avisar que esse treinamento está sendo gravado. Eu acho que veio a informação antes do início, mas só para avisar todo mundo. Obrigada. Lada, you can continue, please. Thank you.
Thank you very much. So thank you, Chrissie, for the oversight, and thank you to the organizers for the opportunity to present Roche case studies. Now that you have heard the different types of complex innovative designs from the presentation that Chrissie gave with the FP position paper, we'll move to present specific Roche studies that have applied complex innovative methods. Next slide, please. We'll start with 2 early phase trials with platform, umbrella, and adaptive components, one in oncology and one in hepatitis B virus. We'll then follow with a histology-independent platform study that uses enrichment and innovative comparative evidence approaches. And we'll end the case studies presentation with 2 examples of innovative statistical methods in late-stage trials. We'll also share some opportunities reinforcing the message that Chrissie has already given, and then we'll hand over to Nikki to talk about dynamic powering. Next slide. So the studies will be presented by representatives from the study teams, and they will go over the rationale for the design itself, and key considerations. Each speaker will focus on different considerations to ensure we cover all of them. As you have seen from Chris's presentation, there are many to consider. And with that, over to you, Jan, to present the Morpheus platform.
Thank you, Lada, and thank you for the nice introduction and the opportunity to present the Morpheus platform in this meeting. Next slide, please. Yeah, next slide, please. So Morpheus is actually consisting of 9 different unique study protocols covering 9 indications. This was actually, the platform was designed to generate quick signals in the cancer immunotherapy combination space and across obviously different indications. This is a multi-tumor type, as I mentioned, but also multi-basket platform, as each of the protocols can consist of different clinical settings, be it metastatic, first line, second line, biomarker defined, or even neoadjuvant setting. Each study is a randomized study having a shared control arm and then consisting of different experimental arms. And it's also kind of a longitudinal platform, as upon PD, a patient has the opportunity to reconsent and then be assigned to a second experimental treatment combination. And last but not least, it's an adaptable platform. All the studies are like strictly aligned and consisting of a core protocol, while the experimental arms and the controls arms are actually covered by appendices that are also quite aligned with regards to working, and then that can quickly swapped in or removed depending on safety and efficacy signals or upcoming promising clinical preclinical data. Next slide, please. This slide is to illustrate you which indications are covered, and here you can see that we are mostly working in a metastatic setting, first to third line setting, but we also have studies that are covering the neoadjuvant setting, as you can see here in the bottom left, the Morpheus bladder study. And we are also working on new additional studies that also cover the neoadjuvant setting here. Next slide, please. And we can go right to the next slide. Here you can see an overview on the key protocol elements. The primary objective of each of the protocols is efficacy and safety. Each protocol is actually kind of an umbrella study design with a master protocol. And each protocol has a length of approximately 503 pages. Of which we have a 100-page core protocol, which is actually, quite consistent with other phase 1, phase 1b studies, and the rest is distributed across the different appendices. Next slide, please. Here we can see like a figure of how these studies look like. You can see that each study, be it in the metastatic or in the neoadjuvant setting, consists of different phases. So we have a screening phase where all the screening assessments are being performed that ends with an entry biopsy. Once a patient is deemed eligible, then he or she can be randomized to either the control arm or one of the experimental arms. Of note, we have also like kind of an adaptive randomization, so we can kind of work with the randomization ratios. As also mentioned, we have a shared control arm here for each study. Stage 1 is actually consists of 2 phases. We have a preliminary phase where we enroll 15 to 40 patients. After enrolling this preliminary phase, we have a formal interim analysis. And if an arm meets the pre-specified gating criteria, we can then decide if we to go to expansion phase and enroll another 25 patients. As also mentioned before, upon progression of disease, the patient is receiving like an on-treatment or re-entry biopsy and can then consent to receive another combination or experimental combination. And this is obviously different in the neoadjuvant arm because their stage 1 and the study ends with surgery. Next slide, please. This slide is meant to show you how one of these indications and unique study protocols can evolve over time. So we, as mentioned before, we can have different clinical settings, and if we look at the randomized part, a study can, like a study for cohort Y, started with 4 experimental combinations, with the first 2 combinations being halted, stopped after the preliminary phase, for example, due to lack of efficacy or due not meeting the gating criteria, while combo 3 and 4 are being expanded as they met the pre-specified gating criteria. Combos 5 and 6 have been added during the course of the study based on, for example, promising early clinical data, promising clinical data. And as mentioned, adding a combo here means adding another appendix to the protocol, so that can be done very quickly. Next slide, please. Obviously, safety, ensuring patient safety is key in these studies. There are actually a couple of challenges. We are working here with combination therapies with up to 4 drugs, which may result in increased drug safety risks for the individual patients. Also, the studies at the drugs enter the study at different phases of their development. So the earliest phase would be like that we have a recommended phase 2 dose, but we also have study drugs that are actually already approved. Obviously, there are different degrees of information for the safety profile, and working with combinations, this can result in potentially overlapping toxicities. We have a couple of workarounds for these safety challenges, I would say. First of all, we implemented in our studies a so-called 6-patient safety run-in. So after enrollment of 6 patients and following them up for at least 2 cycles, we halt enrollment into the arm. Then we do an in-depth review of the safety data, present this to our internal monitoring committee, and then decide if we, for example, continue with the study arm, discontinue the study arm, or would have to adapt this study arm. We are also doing real-time continuous safety monitoring here, focusing on adverse events that are, for example, leading to death, obviously also on SAEs and AEs leading to drug discontinuations. Besides the study teams, we have our internal monitoring committee, which is actually reviewing the automated outputs for each study every 3 months. and we have every 6 months a review of the study data by our scientific oversight committee, which is comprising 3 independent experts. Next slide, please. We have had a couple of regulatory learnings during the course of the study. At the time of the initial CTA submissions, there have been a lot of meetings with health authorities worldwide and a lot of discussions. And we really had to adjust the study protocols and design as per feedback, which was actually quite, as mentioned, an intensive discussion, but it was worth it because Morpheus is now approved in 13 countries across the world, and there are actually a couple of workarounds even with countries that do not have dedicated processes for these platform studies in place. Next slide, please. With time, more and more health authorities became more and more familiar and also confident with this design, enabling increased flexibility of this platform study, adding new indications, adding new arms, removing arms that do not meet our gating criteria, and just to mention 3 examples. So within our gastric cancer study, after discussions with the health authorities worldwide, we added an esophageal cancer cohort for the reason of these 2 indications not being very different. Within our breast cancer study, we added 8 new arms plus a new indication in 1 amendment. Also in our lung cancer study, we added a biomarker selective arm, and this went all quite smoothly because we had, as mentioned, we had a grown confidence into this kind of study platform, and we also shared a lot of information ahead of submitting the protocol amendment with a health authority. And that's it for the Morpheus lung study. I think I can now hand over to Ruchi.
Thank you, Jan. Next slide, please. Thank you. So first of all, I would like to thank the organizers for giving this opportunity. Today, I will be presenting an example of a platform trial to treat infectious diseases, and it is the Piranga trial in chronic hepatitis B. Next slide, please. A brief outline of my talk. Firstly, I'll go into the consideration for the design, why we chose this design, and then I'll move on to the elements of this adaptive platform design. Next slide, please. To give you a bit of background on the current available treatment options for HPV, single agent treatments like nucleoside, nucleotide, or PEG interferon, they are able to maintain the viral suppression in HPV. However, once you stop the treatment, the viral rebound normally occurs. And typically, you need a longer treatment durations to achieve a very small level of functional cure, about 5%. And functional cure is the ability to maintain control over your disease once the treatment is stopped. So the current strategy in the field of HPV is to test a combination of drugs with the different mode of action, mechanism of actions, with or without a standard of care treatment to hopefully trigger a functional cure, and later in the long term maybe be able to find a complete cure for the disease, that is complete reversion of the infected status.
So with this background and Roche portfolio of new molecular entities, NMEs, the objective of this phase 2 was to assess multiple combinations in the best possible way to achieve rapid patient benefit, because it's a high unmet need, and also to accelerate scientific understanding of these new different mechanism and their effect on the disease. Next slide, please. This here is a pictorial view of the functional cure strategies that I just mentioned. So the idea here is, if you look on the left-hand side of this this picture, the idea is to target different stages of viral life cycle using these NMEs with different mechanism of action to, on one hand, achieve viral suppression. So if we now move to the right-hand side, so viral mediated suppression, and simultaneously enhance HBV specific immune response to hopefully achieve a functional cure. Sorry. Next slide, please. So based on the objectives of the phase 2, these were some of the questions that needed to be answered for the design. And moving on, next slide, please. We looked at the traditional design model. So as you can see, like in a traditional design model, if we wanted to assess all the combinations that we wanted to assess, we would have had to design multiple, run multiple studies. If we wanted to test for shorter durations, again, like new studies would have to be instituted. These studies would have had 1:1. Normally, it could be less, 2:1 also, or other ratios of experimental to control. And these would have actually slowed down the development timings. So we wanted to look for a nimble approach for development, and then and we decided to leverage the experience from the oncology, where they have been using combinations to treat cancers. However, acknowledging that there is a very different benefit risk ratio between the two disease areas, simply because of the complexities of oncology. Thank you. Can we move next slide, please? Again, next slide, please. I'll go into the elements of design now. So this is these are the key protocol elements. So our primary objective was to estimate the effect of enemy combination therapies to induce a functional cure over the control arm. Next slide, please. Next slide, please. Yeah, thank you. This is the Piranga adaptive platform design. And I would like to mention here that before finalizing this design, we did seek FDA and EMA scientific advice into the design. So as you can see in this platform, multiple combinations are being investigated in a single study, and these are depicted by the different color bars. And this allows us to answer multiple questions in one protocol, meeting our main objective of rapid scientific understanding. This is also adaptive in the sense we can flexibly, in a data-driven way, we can open or terminate different drug combination. As and when any new treatment becomes available, it is evaluated for its scientific merit to be included in the phase 2, and then we can open different combinations with that treatment. The pre-specified interim analysis in this design help inform incoming combos, and this gives us an early readout on safety, which is very useful for combinations which we have with immunomodulators, as it de-risks the uncertainties sometimes. And interim analysis data also allows us, in case if needed, we are able to open arms with maybe with some staggered in a staggered way rather than opening all the arms with a particular treatment simultaneously. Okay, and as you can see, we have shared control arms, which is depicted by the gray bar at the bottom. And this makes it also attractive for our patients as they have more chances to be on the active drug. And this arm also provides an element of randomization. This trial is also multi-stage, combining phase 2A and 2B. So based on the emerging data, arms are flexibly added if there is a rationale to test different doses. Next slide, please. I will focus on the third bullet point here because I think I have covered the first two. So for this platform trial, we also have one master protocol, which is supporting different combinations. The main body with common element remains the same, and new combinations are added through appendices and as separate appendices and through protocol amendment. And here I would like to emphasize that each amendment is only implemented once the regulatory approval is given. And despite COVID challenges, we have had a very good response to our protocols. So if we can move on to the next slide, please. Sorry, I also want to mention, so we have this efficiency with sites and patients where sites are now familiar with the protocol study design. So whenever we add a new arm, there is less learning curve and there is quicker patient randomization because the timelines for setting up a new arm is very much reduced. And actually, this is the same with the regulatory reviews. Once regulatory authorities in a country become familiar with our protocol, the review of amendment is relatively, we have discovered that it is relatively faster, as only an amendment needs to be reviewed in detail. And of course, this is a complex multi-arm trial, so we had to really streamline our trial infrastructure, logistics, also data standard, data collection, cleaning, and other standards. So this has saved us on operational time and also optimized the cost, which we are able to reinvest in the study to answer even more of our scientific questions. Next slide, please. Some of the challenges here, of course, in the field of HPV, we were not really, we did not know how the acceptance of platform design will be by health authorities and ethics committee. And as such, early engagement, as I mentioned earlier, like seeking scientific advice from EMA and FDA was really helpful. Also, it came as a nice experience for us that although we got some very in-depth, insightful questions on our protocol, the health authorities were very engaging with us and really helped us to help them basically to review and understand our scientific rationale on various aspects of this protocol. And despite the COVID challenges, as you saw on the previous slides, we have had quite a good acceptance from different countries. Next slide, please. And of course, sorry, if we can go back, I just wanted to mention, yes, of course, it was much more intensive to set up, and in non-oncology therapy area, we do definitely need to develop a bit more experience in setting up these kind of trials, as our experience has been, because mostly we have been doing traditional clinical trials outside of oncology. Next slide, please. Next slide, yeah. So this is my last slide. It's basically just a summary of what I have introduced before, just some pros and cons. So I will leave this slide for you to read in your leisure And I think this is my last slide. So I will hand over to the Tapestry trial. Todd.
Thank you, Ruchi. So if we can move to the next slide. And over to you, Todd, to present Tapestry.
Thank you, Ruchi and Lada. I will first off apologize to the ANVISA colleagues. I'm unfortunately unable to get my camera to work today, so I will just be speaking to you via voice. I am going to talk to you about the Tapestry clinical trial, which takes it is a very complex trial, but takes a new pathway of histology-independent approach for study in oncology. Next slide, please. So as was discussed by some of the previous speakers, the advances in science have really led us to a new space and also created some challenges for us. The great work done by the Genome Project has allowed us to identify somatic alterations in many diseases, inclusive of cancer, that underlie the cause of the disease itself, and gives us a greater understanding of what is otherwise very heterogeneous diseases. In the case of oncology, it allows for targeting of essentially what is considered subpopulations of tumors previously defined by the tumor site of origin, but now perhaps we can redefine that based on our understanding of the disease and the driver mutations and other biomarkers involved in the oncogenesis process. The improvement in technology allows us to not only identify these alterations, but also to essentially identify these patients who have these alterations and to develop drugs that can specifically target these alterations, as opposed to less specific therapy such as chemotherapy utilized in the past. It leads to perhaps more homogeneous populations, more similarly identified based on the alteration. However, these are essentially smaller populations, particularly when we focus within a particular traditional tumor site of origin. We can develop these drugs and identify these patients. However, with the narrowing of the populations, this presents some challenges that we must overcome with both trial design and statistical approaches. Next slide, please. So when we look at precision drug development and how Tapestry will fit with these, the methods of obtaining supportive evidence for these rare populations are challenging within the context of a traditional trial. The Tapestry trial, as I'll show you, essentially is a platform trial that is essentially like an umbrella of baskets, permitting us to obtain enough information across various tumor types with a particular alteration to show evidence of benefit for these patients. However, the limited nature of the population size mandates that single-arm trial types of approaches are taken within the study. And alternatively, our team has thought about how can we supplement those data with the use of real-world data and other unique analyses within the trial itself to answer some of the questions that would remain in the absence of a randomized clinical trial. Next slide, please. So to go into the Tapestry trial, some of the goals and the rationale for the Tapestry trial, as mentioned, the genomic alterations are often too rare to study within tumor histologies or tumor sites of origin themselves. And so this histology-independent approach allows us to enroll enough rare molecularly driven patients to assemble a data set and to evaluate the efficacy of targeting that particular alteration. It does lead to a single arm trial design for each cohort, and we can demonstrate the activity with an ultimate intent to prepare for comparison to standard of care. And that can be done both within the trial, but also with the use of the real-world evidence that I alluded to. Genomic testing ultimately is not consistently conducted. It's something that's growing in its utility, and often for rare alterations, such as those that we're studying, it may not be justified, particularly within individual tumor types where the standard of care may be perceived as more general. We've addressed this through a number of mechanisms, one of which is by assembling enough different drugs and alterations which are justified for study in a tumor agnostic way or a histology independent way. The testing is justified. We're also allowing the use of local high quality NGS testing to permissibly find these patients. Central blood-based testing may be utilized only in the cases where patients do not have sufficient tissue. It's primarily based on a tissue-based approach. Additionally, drug development in pediatric populations has always been a challenge. We heard an allusion for use of extrapolation methods for pediatrics. In this case, because these alterations do also appear as driver alterations in pediatric tumors, it affords us the ability where the dosage formulation and dosage recommendations can be put forth, and we have identified that the alterations exist in pediatric tumor types to study them along with the adults and assimilate the data in totality to support not only a tumor agnostic but potentially an age agnostic effect of the drug by targeting the alterations. Next slide. Just how rare are these? These are some examples of some of the various alterations that could be considered for study in a tumor agnostic fashion, and you can see that some of the alterations are quite rare across all solid tumors. If you look, as things get to the lower portion of this, it may appear that this is a fair percentage of patients across tumors. However, within individual tumor types, there's still an insufficiency to identify enough patients to conduct traditional trials, and therefore the tumor agnostic approach is the best approach to study this. Next slide, please. So the study itself is classified as a phase 2 study, partially because of the potential for registrational intent, not being able to conduct this as a phase 3 study. I mentioned that this is a platform study. It's essentially like an umbrella of baskets. Because each arm is single single arm and not randomized. Overall response rate is declared as the primary endpoint. However, we are collecting some of the more traditional durability endpoints for the use in comparisons within trial data as well as across in comparison with real-world evidence. The study, similar to some of the others, is quite large in length, the first version being 431 pages, and that is growing as we amend the protocol and add additional arms. Similar to what some of my fellow speakers alluded to, the study is set up with a basic main body protocol with annexes or appendices for each cohort, which can be added or subtracted as the study goes along, and does facilitate, hopefully, consistent and easier review with subsequent additions. Next slide, please. The study itself, as you can see, currently has 10 arms in version 2. As mentioned, these patients are across all solid tumors and tumors that can be assessed by RECIST criteria for a measure of consistency in most cases. These patients should have either progressed on prior therapy or for whom there is no acceptable treatment available. This does permit that the clinician would choose when the appropriate time frame in treatment is for an individual tumor type and an individual patient. As mentioned, we allow enrollment via local testing, predominantly tissue-based testing. In the case where a patient has an insufficient tissue for testing, we do permit that a specimen be submitted for blood-based testing for identifying these patients. And because this is a modular platform study, it does allow for the addition of future arms when scientific rationale justifies the study in a tumor agnostic fashion. And we are continually looking for more targets with which to enhance the efficiency and utility of the study. Next slide, please. So one of the most unique elements of this, aside from the histology-independent approach within the trial, is the fact that we recognize that both health authorities and HTA assessors and payer groups would like to see more comparative evidence, despite the fact that the rarity of these populations and the unique nature of histology independent makes that challenging. So we are not only collecting data in the trial, which you just saw, on the patients who are treated with the drugs that are under study, but we have intentions of assembling essentially a total data package with the idea of addressing most of the clinical questions that would be addressed in a randomized clinical trial to permit a benefit risk assessment for each of these drugs. And essentially things like an intrapatient comparison to last prior therapy that these patients received before being on the trial will help speak to any potential considerations of bias of the patient enrollment, seeing that some of these patients clearly had progressed or not benefited from prior therapies indicates that it isn't just a good prognosis population that does well with each therapy. As well as amassing a large external real-world database will allow elements of defining natural history of the standard of care, the prognosis of the biomarker, and ultimately potentially early for registrational purposes and later for continued reimbursement assessments, comparison particularly on some both traditional and non-traditional elements of durability of effect beyond the primary endpoint of response rate in the study. Next slide, please. And so the umbrella and platform trials really are an innovative methodology, as alluded to earlier, and the main focus for us is a mechanism by which we can enroll rare patient populations. The tumor agnostic approach takes that one step further and allowing to enroll patients across tumor types that are similarly defined by the presence of an oncogenic driver, to allow study where it would otherwise be feasible within individual tumor types. The use of both tissue-based testing and ctDNA ultimately allows for the potential for identification of all patients who possess these rare oncogenic driver biomarkers, and amassing real-world data for more patients treated with standard of care therapy allows for more closely matching to the trial population and relative comparison of durability of effect endpoints. With that, I would like to conclude and hand over to my colleague Kaspar to talk about some innovative statistical methods.
Thank you, Todd. I just wanted to remind everybody, the attendees, that you can send questions via the chat and the Q&A. We'll be happy to respond afterwards. So thank you, Todd, and over to you, Kaspar.
Thanks, and thanks for the opportunity to present two actual phase 3 clinical trials where we think we have used some innovative aspects, mainly on the statistical side. Next slide. I would like to start with CLL11 and would like to present that as an example of a statistical innovative technique that we used outside of the list that Chrissie actually presented. And to understand why this was innovative, let me briefly introduce the objective that we had in this trial and the scientific question that we tried to answer. In CLL, at the time when we planned this trial, there was an approved standard of care, chlorambucil. And we wanted to assess whether Gazyva, a second-generation anti-CD20 antibody, combined with chlorambucil, actually would do better on a primary endpoint of PFS. But the challenge was that the first generation anti-CD20 antibody, rituximab, had never been approved in this indication. So if we just compared Gazyva plus chlorambucil against chlorambucil, and that assumed that trial would have been successful, a lot of questions would have remained open. Would rituximab also be already better than chlorambucil alone? Is Gazyva which we claim is a second generation anti-CD20 antibody, actually better than rituximab? I think these would have been fair questions. And if we just had compared what is here given as B versus A, these questions would not have been answered. On the other hand, we wanted to move quickly with this molecule, of course, and bring it to patients fast. So running 3 trials, looking at all combinations, was not really an option. So how can we bring in statistical innovation to answer all the questions we have, but still not dramatically delay timelines? That was the kind of the scene that we met, and here's what we proposed and what we actually did. Next slide, please. Next slide. So we have 3 comparisons that we want to perform: chlorambucil against Gazyva, chlorambucil against rituximab, and Gazyva against rituximab. How can we do that efficiently? The next slide, you will see the answer. The statistical method that you can use here is closed testing. Next slide. So with closed testing, before you do your first pairwise comparison in a 3-arm trial, so you put all 3 therapies in a 3-arm trial, and before you do your first pairwise comparison, you do a statistical hypothesis test that looks at all 3 arms at the same time. And I spare the technical details. If you pass that test, you can actually perform all the pairwise tests at a significance level of 5%. So the correction for multiple testing is induced through that global test you do prior to the first pairwise comparison. And you see here how that actually compares to potential alternative options that we could have taken to answer our scientific objective. We could have run 3 separate trials. So A versus B, chlorambucil against Gazyva, chlorambucil against rituximab, and then I think especially payers, they wanted to know, is Gazyva actually doing better than rituximab in this indication? So we would also have needed a trial B versus C. But then you need to recruit patients for all 3 arms in these trials. You are not kind of recycling the patients from the chlorambucil arm for both comparisons. Okay, so you need chlorambucil patients in all of these 3 trials. So recruitment and will take a long time. You need a lot of patients if you run 3 separate trials, of course, and that leads to a long time until clinical cutoff. So you see it here. If we would have run 3 separate trials, the clinical cutoff, or the time until analysis of the first comparison, chlorambucil versus Gazyva, would have been 34 months. Okay. In the second trial, comparing chlorambucil against rituximab, the time to the clinical cutoff would have been 39 months. Okay. Can you please go back 1 slide? If a naive approach to this problem would have been that you put all the 3 arms into 1 trial, so not run separate trials. The benefit of that is that you can use the chlorambucil patients for the comparison to Gazyva and to rituximab, because they're all in the same trial. But one way to account for the multiplicity, for the fact that we actually run 3 comparisons, would have been to just do a Bonferroni correction. So you take the overall significance level, 5%, divide it by 3, and then plan all your trials using that significance level. Because you have this more stringent significance level, 0.05 divided by 3, that increases your trial size. So, but here you would already have gained, and that's because mainly because you are reutilizing your patients for all the comparisons. You are not having them in separate trials where you need to recruit chlorambucil patients into 3 trials or 2 trials. So here you bring down the time to the clinical cutoff to 21 months. So you see here already quite the gain compared to running separate trials. Now, if you do close testing, you can even improve on Bonferroni, and you see here by how much you can actually improve. You again run a 3-arm trial, but you use a different, a more innovative correction for multiple testing if you want. And then you see here the time until the to the cutoff for the main comparison of interest, chlorambucil versus Gazyva, can further be reduced to 18.6 months. But the main benefit here is that you see that mainly for the other comparisons, we actually get realistic timelines. Here, these timelines would have been completely unrealistic with using these approaches. But with the closed test, we see that even for the most challenging comparison, there where the relative effect was smallest, namely Gazyva against rituximab, you see here the hazard ratio that was assumed for powering, about 0.74. We can still get a realistic timeline until the clinical cutoff. And here we again gain 5 months for the comparison chlorambucil against rituximab. So I think the take-home message here is that even outside of the list that Chrissie presented, that we currently consider innovative designs, there are always statistical methods that can be used to improve on more kind of template approaches that we often use. And here I would like to emphasize this is all. So this is a method that provides full type 1 error control. So there's no cheating. This is all according to guidelines and how we are supposed to run trials. It's just using a very efficient fit for purpose method for the scientific objective that we had in this trial. Okay, and if you're interested to get some more details, this is the clinical publication, Good et al. And this is a methodological publication discussing the statistical methodology, Asikanius et al. Okay, next slide, please. My second example is an example of Chrissie's list, an enrichment trial. So that's one innovative aspect. And the other innovative aspect I would like to emphasize is that this enrichment design was put into this trial only after the trial had already started, based on external evidence that emerged. And I would also like to showcase how this can be done without compromising the actual integrity of the trial. So Impassion 031 is an enrichment design in immunotherapy for a binary primary endpoint, pathological complete response. And the trial was originally designed in triple negative breast cancer in a non-adaptive way, a simple 2-arm randomized clinical trial comparing atezolizumab plus chemotherapy versus chemotherapy alone. The trial was fully enrolled, so 205 patients were already enrolled, when external evidence emerged hinting that PD-L1 status could also be predictive of the treatment effect, so that we might have a differential treatment effect in the subpopulation of patients that have a positive PD-L1 biomarker expression. Now, should we take that into account? Because this is something scientifically we would be very much interested in. There is also the risk that if this is really predictive, that the effect in the complement, so in those patients that were recruited into the trial but were not PD-L1 positive, could actually not sufficiently benefit. And it would be a pity that if the result in those patients that would not benefit would kind of compensate or invalidate the trial in a sense that it would not be statistically significant. So how could we, or what is an option to react to this emerging external evidence? Next slide, please. So the approach we took is to make this into an adaptive enrichment trial. And in an adaptive enrichment trial, the 200 patients that has already been recruited, we would not perform the final analysis as initially planned, but we would use these patients to either decide what hypotheses we will test at a potential additional final analysis with additional patients recruited, namely whether we would use that data, or rather we would present that to an IDMC, and then IDMC would make that recommendation. Should we, at a postponed final analysis, only look into the subset of the targeted PD-L1 patients? Should we not bother about the biomarker? So this was external evidence, and it was unclear how that applies in our trial in the 205 patients that we had already recruited. So should we not bother about the biomarker, or should we actually look into both populations? Should we test at the final analysis in the full triple-negative breast cancer patients, targeting some effect? Maybe there we see a hazard ratio of 0.75, but potentially we see an even stronger effect in the PD-L1 subpopulation. Okay? And you would use the already recruited patients to make the decision what you would look at the final analysis. And this is what has become known in the literature as an adaptive enrichment design. Again, I would like to emphasize that this has full control of type 1 error, if properly set up, of course, also if using the proper statistical methods. And all these modifications, they were performed based on external evidence. That means before unblinding the trial, and all these modifications or the recommendations, what we should look at the final analysis, were done in a matter such that we as a sponsor remained blinded. So this was an instruction to the IDMC what they should look at and what recommendation they should provide. And this allowed to kind of repurpose those 205 initially randomized patients and repurpose the trial to answer an updated scientific objective, if you wanted, maintaining the full integrity of the trial. Again, the clinical, and maybe I can share the results. So eventually the trial was statistically significant in the full patient, or in the full population of triple negative breast cancer, and was just not significant in the PD-L1 subset. One should acknowledge that, of course, if you look in a subset, you need quite a large treatment effect because the subset is smaller than the full population. So you will have more variability. If you want to become statistically significant, you need a quite substantial treatment effect. Again, the clinical paper is given here, and the methodological paper where this design is described as well. And with this, I conclude, and I think we are all more than happy to take questions. Back to Lada.
Thank you, Kaspar. So with that, I have one more slide, and then I will give over to Nikki. Next slide, please. So now that you have heard the designs of the case studies that from the Roche trials, I wanted to look back at the opportunities that these innovations offer, reiterating the message that Chrissie gave before, and also the speakers over the different case studies. So as you have seen, we can elucidate robust evidence through strong scientific rationale and methodology. We can drive patient centricity, and we can learn, we can have higher efficiency, as we saw before, through having answers to the scientific questions earlier. And as you have seen as well, in line with good clinical practice and ICH guidances, patient safety and data integrity remain our highest priority in these designs. Now, from my previous experience as clinical trial assessor, I know how challenging the review of these studies can be. So I wanted to share with you 3 thoughts that made it easier for the assessors at Swissmedic when I was working at the agency. So as some of my colleagues shared, having pre-meetings to discuss study designs before the CTA submission, and if necessary, also having calls before amendments, helped to much better understand the rationale for the design and its components. Also, depictions of the study in diagrams, like you have seen throughout the presentation, can be really helpful for the assessor, especially when amendments are submitted. And one last thought I wanted to share, that even if master protocols seem more complex, they can be broken into pieces that fit the usual review process. And this is how we approached it at the agency back then. And also, as some of my colleagues shared, having the when agencies are reviewing the amendments, it goes much smoother because the main protocol components are already known. So with this, I hand over to Nikki to share on dynamic borrowing.
Thank you, Lada, and thank you to the organisers for the opportunity to present at this training. We move to the next slide, please. Next slide. Thank you. Sorry, can you just go back one slide? Thank you. So I'm going to present on a slightly different topic, talking about use of Bayesian dynamic borrowing, statistical approach to bridging studies. And this example, I'm going to present two case studies from GSK. These are both examples. In Chrissie's list, you mentioned use of historical controls. So this is a related example using historical or external data, but actually using external data directly about the treatment effect of interest rather than just on the control arm. To say borrowing in the treatment effect in the context of a bridging study setting. So next slide, please. So bridging studies or multi-regional clinical trials can be thought of as a form of evidence synthesis. So decision-making around the target region or population is based on the totality of evidence about the overall treatment effect, and then evidence around the consistency of treatment effects across the regions or subpopulations of interest, and consideration of what's known about intrinsic and extrinsic factors that may impact treatment effects across different regions and subpopulations. Next slide. So the formal synthesis of data from the overall population and the data in the target region or population of interest using statistical modelling approaches can help inform regulatory decision making about regional or subpopulation treatment effects. Next slide. And of course, one widely used method for carrying out evidence synthesis is meta-analysis. So when we have several subgroups or regions of interest, then one way to synthesize that evidence through a statistical model is to conduct a meta-analysis. Next slide. So a related idea to meta-analysis, another approach to evidence synthesis that's been gaining a lot of interest is this idea of reusing or borrowing data from an existing study to supplement or add to the information collected in a new study. And you can perhaps think about this idea of borrowing data as sort of adding virtual patients into the new study. So if we imagine we have a target patient population of interest, next slide. Sorry, I've got several builds here. So then we have a source study conducted in that population. So this is a little cartoon to illustrate we've collected some data from this source study, and that provides us with an estimate of the treatment effect indicated by the histogram. The dotted vertical line is your estimated treatment response in that source study, and the double-headed arrow gives you an indication of the precision, the confidence interval for that treatment effect. Next slide. So if we imagine that we now conduct a new study in the same target patient population, we collect a new set of data and a new set of patients, and on the right you can see another estimate of the treatment response in that population based on our new study. Again, the dotted line is our estimate with the double-headed arrow giving an estimate of the confidence interval for that treatment effect. Next slide. And then the idea of borrowing data or adding virtual patients is very simply in this cartoon, we take the source study data, we add it, combine it with the data collected in our new study, we end up with our estimate in the bottom of the treatment response in that target population based on the totality of the new study data plus the data we've borrowed from our source population. So that just gives a very high-level illustration of the idea of borrowing data from a source population or from a source study. Next slide, please. And so we can operationalize this idea of borrowing data from a source study into a new study through statistical methodology using Bayesian statistical methodology. We can think of the source study data as being our prior information, which we combine with the data we collect from the new study, and then the combined estimate of the treatment effect that we get is called the posterior distribution. So that's an application of Bayesian inference. Next slide, please. Now, Bayesian inference can be used in many settings, using the source study data as the prior information, but the posterior estimate, the updated estimate of treatment effect we get in that setting is based on pooling the source data and the new study data. Next slide, please. The key question arises, though, in terms of what if the source data and the new data are not entirely similar. Now, we would never expect necessarily to get identical data from two different studies. There could just be differences due to sampling variability, but there may also be real differences between the source population and the target, the new study population. So there might be, for example, differences in the inclusion-exclusion criteria between the source and the new studies. There may be differences in the sort of standard of care, the health management of patients in the different settings where the source study was carried out and the new study. There may be differences in the baseline risk factors and other aspects of the populations which differ, all of which could lead to real differences between the treatment effect in those source and new studies. But nonetheless, they may not be entirely different. They may still be useful information from the source data that's relevant to assessing the treatment effect in the new data. So the idea of Bayesian dynamic borrowing is to enable the opportunity to borrow data from a previous study or studies, but to allow or acknowledge the possibility that there may be some drift, some difference between settings, and so we want to account for that possibility. Next slide, please. So the idea of dynamic borrowing is that the larger the evidence of drift, the larger the differences between the source and the new study settings, the less we want to borrow the source data, and vice versa. The smaller the evidence of the drift, then the more we want to borrow when we're making inference about the new study. Next slide. So just a little cartoon to illustrate how this dynamic borrowing works. So again, imagine we have our source data that we've collected, providing an estimate of the treatment effect. Next slide, please. And one thing we need to do when using dynamic borrowing is we need to provide an initial assessment of how relevant or similar we believe the source data is likely to be to the new study setting. And so in this cartoon of this, we've made an assumption that we put a 50% initial weight, sort of 50-50 belief that the source data is relevant. Next slide, please. We then conduct our new study and we collect some data in the target study population. And if there's little or no evidence of drift, the data in the source and the target study are very similar, then the dynamic borrowing works by actually updating the weight that we place on the source data, and that increases. So actually we end up borrowing more data because we've got evidence of similarity, and so we can borrow more information to support the estimate in the target study. Next slide, please. However, suppose that the data we observed suggested some moderate drift between the source data and our target population. That may suggest that there's still some overlap, there's still some relevant information, but we may not want to upweight it quite as much. So now we only upweight a little bit to say 55% weight we're putting on the source data. Next slide, please. And as if in another setting we actually observed much larger drift between the target data and the source data, then actually we would end up downweighting the source data even more than we started with, and so that might go down to only putting a 5% weight on the source data. And next slide. If we observe a sufficiently large drift, then actually the dynamic borrowing effectively completely discounts the source data, and we only base inference in the target study on the observed data from that study. So you can see that depending on the evidence of drift that we observe when we collect the new study data, we can either upweight or downweight the source data relative to the initial weight that we placed on it. Next slide, please. Just to summarize then, so the dynamic borrowing requires this initial judgment, which I'm calling the prior or initial weight, on the likely relevance or the consistency between the treatment effect in the source data and the target population. Next slide. But given this initial weight, then the final amount that we borrow is data driven. And the bigger the observed drift or lack of consistency, the less information we'll borrow to boost the new sample size and vice versa. Next slide. And it's important to emphasize that this upweighting and downweighting is data driven. We're not changing the prior partway through the study in a post hoc way. This is built into the prior and the statistical methodology before the study starts. So this adaptive element could be pre-specified, and as I say, is driven purely by the data. Okay, next slide, please. So I'm just going to give you two brief examples to illustrate how we've been using Bayesian dynamic borrowing in a bridging study setting. So the first is actually a retrospective application where we've used this approach as a supportive analysis. And actually, this is something that at GSK we've used in several pediatric regulatory submissions to support partial extrapolation of efficacy from adults to children. And a similar approach could also be considered in sort of bridging studies, maybe bridging between different regions or subgroups. Next slide, please. So this example, first case study is a retrospective use of dynamic borrowing to support pediatric approval of Benlysta for pediatric lupus. And this was analysis that we carried out at GSK in response to a supplementary information request from the FDA during the review of the pediatric label extension submission. And actually, the FDA then presented this methodology in this example at the American College of Rheumatology conference in 2019 as an example of innovative use of Bayesian methodology to support pediatric extrapolation. So next slide, please. So to illustrate how this worked in a retrospective setting, this slide shows you the data collected from the adult study on the right. So this was the adult phase 3 study. The primary endpoint was a disease activity index. So the odds ratio of being a responder on Benlysta versus placebo. So in the adult study, there was about a 1.5 odds ratio in favor of Benlysta, which was highly significant. In the pediatric study, we observed suggestive evidence of a similar treatment effect in pediatrics, but due to the very small pediatric sample size, due to the rarity of lupus in pediatrics, the confidence interval was very wide and didn't meet conventional levels of significance. So the FDA requested an analysis which formally incorporated the evidence from the adults to help assess the evidence collected in children, given the similarities in the disease management and pathophysiology between adults and pediatrics, and the similarities shown in the PK for Benlysta between children and adults. So next slide, please. So what we did was to carry out a series of analyses using dynamic borrowing, where we varied the initial weight that we placed on the adult data, varying from the left, putting 0% weight, which effectively is ignoring the adult data, all the way up to the right, where we put 100% weight on the adult data, which is equivalent to pooling the adult and the pediatric data. And the idea was to identify the minimum initial weight that would need to be placed on the adult data, such that the combined estimate of the treatment effect in children, having combined the adult and the pediatric data via this dynamic borrowing approach, met conventional levels of significance. In other words, that the 95% interval excluded the odds ratio of 1. So next slide, please. So you can see here that an initial weight of 55% on the adult data. If you look at the blue line, the estimated pediatric effect, you can see that excludes the odds ratio of 1 once we put at least 55% weight on the adult data. So that was the, if you like, the tipping point, minimum prior weight needed in order to conclude substantial evidence of efficacy in pediatrics. Next slide, please. And so the FDA conclusion was that a weight of at least 55% on the relevance of the adult information to the pediatric population was considered to be a reasonable assumption given the clinical similarities and the PK similarities in this setting, and therefore that this tipping point analysis supported a conclusion that the treatment effect of Benlysta in the pediatric population favoured Benlysta compared to placebo, and ultimately the entire file led to approval of Benlysta for treatment of pediatric lupus. Okay, next slide, please. So the second case study I'm going to show is a prospective application of dynamic borrowing as a pre-specified primary analysis rather than as a supportive analysis. Next slide. So this is in setting of a bridging study to support registration of an approved medicine for a new indication in China. So just a little bit of background, GSK had developed drug X as anonymized the details. So already developed drug X and it's approved globally, including for China for indication 1. But the program is now under development for a second indication, and a pivotal phase 3 study is being conducted to support that second indication. However, China wasn't included in that pivotal phase 3 study, and so the proposal is to now conduct a bridging strategy to support approval of drug X for use in indication 2 in China. Next slide, please. So a sort of traditional approach to this type of bridging or consistency design, either as part of a multi-regional clinical trial or as a standalone bridging study, typically would be to use what's called or referred to as trend analysis. So in this case, a minimum treatment difference would be specified. This would be the difference we would need to observe in the bridging study in China in order to achieve a positive result. So it's not a traditional fully powered study with a conventional hypothesis test. It's a trend design where we need to observe a treatment effect that trends in the same direction as the global treatment effect from the phase 3 pivotal program. And then if the observed effect in China is greater than that minimum difference, then the treatment effect estimate in China is just based on the observed data in China. But due to typically the much smaller sample size in these sort of bridging settings, that will be a very imprecise estimate of treatment effect. So next slide, please. So instead of that sort of trend analysis, GSK proposed to use Bayesian dynamic borrowing as a prospective design to enable us to formally incorporate the evidence of treatment efficacy already obtained in the phase 3 pivotal program for this second indication, and to incorporate that in a formal way to enable the decision sort of hypothesis test to be carried out and to enable an estimate of the treatment effect in China that incorporates that additional evidence from the phase 3 study. So we're effectively adding in the Bayesian dynamic borrowing. So we're formally incorporating the phase 3 data based on the scientific rationale for similarity of the treatment effect in Chinese and non-Chinese patients. We pre-specify a weight of belief on that similarity in the design of the trial. We can then specify a hypothesis test based on requiring 95% probability of a true treatment benefit in Chinese patients. and then combining that with the planned sample size in China, those three elements will then lead to the minimum effect that needs to be observed in China in order to meet the decision criteria. And then if the minimum decision effect is met, then the estimate of the treatment effect in China is then estimated based on the Bayesian dynamic borrowing model, incorporating the phase 3 study data as well as the China data. So we're effectively providing more robust analysis and robust design to evaluate the efficacy in the China population compared to the more traditional trend analysis. Next slide, please. So I'll just leave you, if you can just, next slide to add in the other bullet points. I'll leave you to read these in your own time. This is just to summarize the benefits of the Bayesian dynamic borrowing in this sort of setting. Next slide, please, and some of the challenges. Thank you. And then final bullet point, just to comment that, sorry, next slide, please. That this approach using Bayesian dynamic borrowing has been recognised by several regulatory agencies, FDA, EMA, and the Chinese regulatory agencies for use in these sort of bridging and extrapolation settings when there's a strong scientific rationale for the relevance of the data being borrowed. Thank you. So I'll hand back now to Lada, is that right?
The organizers, thank you, Nikki. So Camila and Luciana, back to you.
Olá, pessoal. Então, agora é o momento, agradecemos a todas as apresentações, excelente conteúdo. É o momento para a gente aproveitar e fazer perguntas. Nós não vemos nenhuma ainda aqui no chat. Alguém, por acaso, enviou alguma pergunta que não estamos conseguindo ver? Então, a pergunta é: How do we apply the bridging trials in order to extrapolate the data from adults to children in the pandemic COVID-19? So I think, Nikki, if you could address that.
Thank you. So I'm not sure if the question is asking specifically around COVID-19. I think it's more generally in the current setting of COVID-19 and the challenges that brings for other trials. I think the example that I gave, bridging retrospectively for Benlysta in lupus, I think a similar approach can be used, and we have used in other disease settings as well. If you have adult data available, then that can be used as a supportive analysis in terms of then providing additional supplementary information around the pediatric effect if you have a pediatric trial. If you don't have any data collected in children, then the dynamic borrowing approach that I used is not applicable. You do need to have some pediatric data collected. So in a setting where you're unable to collect data in children at all, then a different approach would be needed. But if you have some data collected in pediatric trial, then those approaches can be used. Or alternatively, you can design the trial prospectively in children, similar to the way we used the design for the China study, to pre-plan the use of adult data to supplement the data you can collect in children. But I don't see any specific differences in the pandemic setting compared to the examples that I talked about. I think those approaches are relevant in settings where you have got good scientific rationale for similarity of the adults and the children.
A pergunta foi feita por Leonardo, né? Leonardo, se tiver alguma questão a adicionar, por favor, você puder adicionar no chat. Temos uma pergunta também aqui de Gabriel Pinheiro. Em um estudo adaptativo, como funciona a questão de crossover? É possível ou não? So the question is, in adaptative study, how it works, the matter of crossover? Is it possible or not? So anyone can take it?
As Chrissie, so the question I believe is patients could cross over from a particular arm and go on to an active arm. So that's the crossover. So people might drop in or drop out of treatments. Yes, that is possible in adaptive designs. That would have to be a careful consideration in the design. In terms of if for example, in oncology, if someone is to fail the treatment, what do they then go on to? So we look at the treatment sequences and what are the treatment conditions that we are mostly interested in. So that would have to be built into the design if that was the right sort of answer to the right question.
Excellent, thank you, Chris. Any other question? In the same path of my last question, is it possible to apply the bridging studies in order to extrapolate results from different vaccines platforms, considering the ethnicity factors E5, the ICH E5? Who can take it?
I'm happy to sort of
thank you, Nikki.
Take it, but if anyone wants to add, I mean, I'm not sure if there is what specific ethnicity factors. Are you talking about ethics or ethnicity? I'm not sure what the question was, but in principle, I think the bridging strategies that I talked about can be applied. The key factor in any situation needs to be the scientific rationale for the expected similarity. So if there is a good reason to believe that the adults or the population that your source population is similar in terms of the characteristics, the disease management, the PK, the expected treatment benefit in the target population, then the approach is a possible approach. And the idea of the dynamic borrowing is that if there are unexpected differences, then the data you're borrowing can be down weighted. But I think it's a very case specific situation in terms of the rationale for doing the borrowing. The advantage of this approach is that you have to be explicit and transparent around the belief that's being placed on the similarity at the design stage of the trial. But I'm not sure if there are other factors, or if anyone else wants to comment specifically around the E5 question.
Chrissie, all I would add is that as per other studies, we try to generate evidence in different ethnicity factors for example, in a multi-regional clinical trial, we try to get a sufficient sample size in different ethnic groups. So we can at least summarize the data. Sometimes you do need to do some additional bridging studies if you are unable to collect the minimum sample size. But I think with ICH E17 along with E5, it's recognised that we try to pool across regions and in addition to look at the demographics as much as we can to generate the data to demonstrate the consistency of the treatment effects. So again, it comes down to that obligation to try and recruit as wide as possible, both regions and ethnicity as much as possible. So it's a conversation we have with the regulators in terms of the evidence that we have prior to filing.
Great, thank you, Lada and Nikki. I think maybe we have time for one more question. Given the complexity of adaptive studies, is it correct to assume that there will be more protocol amendments? If so, how have major regulatory agencies such as FDA and EMA handle evaluations and approvals?
Maybe we can share the experience from the Roche studies. So there are more amendments in that there are amendments to add new arms or to take arms away, as you have seen through the Morpheus and the Piranga study, as well as Tapestry. How the agencies are evaluating the clinical trial application approvals is by making sure that the new arms or the amendments are in line with the scientific rationale of the main study protocol, and that the annexes that are added with the amendments are still in line with that, and then they would go about with the same evaluation for safety and efficacy, for patient safety and IMP quality as they would do in another more normal study. So that's how agencies that do the clinical trial approval will evaluate this. At the time of marketing authorization approval, it would be about looking at how this data is scientifically valid and contributes to the safety and efficacy evaluation. And as we have shared as well, agencies have been very open to, in the approval of the amendments, very open. And since they understand the main protocol much better, these amendments have been approved very easily.
Okay, so one last one, and then we can answer the additional ones through email. So also for Leonardo, thank you. Is it possible to apply the dynamic borrow approach? Okay, it's a new one. Is it possible to apply the dynamic borrow approach in order to estimate the posteriori distribution in age groups for COVID-19 vaccines? For example, elderly population, considering the low amount of data in this age group. Anyone could take it?
So again, I'm happy to take that if others want to join. So again, yes, I think in principle the approach could be used. It can be used either for separate studies, so when the information you want to borrow comes from a separate study, but it can also be used to look at subgroups within a study. So if you have a vaccine study, or we have data in the same study on different age groups, then it could be used to borrow information from a younger age group to support the estimate in the older age group. So again, that approach can be used, provided there's a good rationale to believe that the effect in the older age group should be similar to the younger one, and you may want to just focus on, not the entire younger age group from 18 upwards, but just say the older adults, sort of 50 to 65 or 40 to 65, so you can pick the appropriate subgroup to borrow from.
Okay, Leonardo answers here for thank you, Unique. So I think it's time to conclude. We are 5 minutes past. I'd like to thank you so much, all the ANVISA attendees and their questions, all the panelists for their excellent presentations. We will be posting the presentations and also any additional questions that you might have. Please feel free to send to Interfarma, and we will contact the panelists to answer to you. So thank you so much. Have you all a good day, good rest of the day, and see you in the next opportunity. Thank you. Bye bye.
Nos módulos anteriores, os speakers se disponibilizaram em abrir um canal, enfim, para tirar alguma dúvida. Eu acho que se eles puderem fazer isso agora, seria ótimo.
Seria bem importante, Cláudio.
Perfeito, Claudio Svan. So Claudio Svan and Solange are telling us that in the other opportunities, there was an open channel for discussions within the ANVISA members directly with the panelists. So if you allow us to do so, we can share any of your contacts to them. Please let us know.
That's fine, it's Chrissie.
Yes, of course. Thank you for the opportunity. That's Lada.
Thank you, Chrissie. Thank you, Lada. Is that okay, Nikki?
I think Nikki's gone, but I will give her. She's fine to share her. She's let me know she's fine to share her contacts.
Okay, So we'll share your contacts with the ANVISA attendees so they can contact you directly in case of any additional further questions.
Thank you, Luciana.
Solange, você quer fazer uma conclusão final?
Obrigada, Luciana. Obrigada de ser a nossa intérprete aqui. Muito obrigada. Bom, eu acho que foi bastante produtivo, bastante denso, um assunto complexo, bem difícil. Eu agradeço muito a presença de todos até o momento e com várias perguntas e talvez ainda não tenha finalizado, a gente abriu a possibilidade de perguntar por e-mail, então agradeço demais. Quero agradecer também a equipe toda que trabalhou para que esse evento acontecesse, Luciana, Camila e várias outras, eu agradeço demais isso, que sem elas isso seria bem difícil que acontecesse. E thank you very much for the international speakers. Thank you for sharing your knowledge with us. It was very important for us. Então, bye bye.
Cláudio Osvan, não sei se você quer dizer algumas palavras finais.
Bom, eu gostaria só de agradecer, Luciana, a todas vocês e toda a equipe da Interfarma, né, que por todo o empenho, né, para fazer acontecer, como a Solange disse, realmente sem vocês não seria possível. E nós estamos muito satisfeitos, não só com esse módulo, mas desde o início, né? Então, acho que eu sempre reforço que nós abrimos aí uma oportunidade, um canal, né? Uma estratégia muito importante para nós. Este, de fato, é um tema muito importante, dos mais relevantes, como eu disse, principalmente agora, né? Com a Covid-19, tem aparecido muito, e a cada um que aparece é um novo desafio. Então, eu acho que a gente ainda tem bastante para aprender e, enfim, né, para avançar, e eu acho que esse evento nos ajudou bastante com isso. Então, eu agradeço novamente por tudo e boa tarde a todos. Tchau, tchau.